In this concept, we will be talking about the phases of the cell cycle, mitosis and the mitotic spindle, cytokinesis, and binary fission. Cells need to control how often they divide. Unicellulars have to divide or reproduce only when there are favorable environmental conditions, such as enough food, for the new cells to survive. In multicellular organisms, cells need to divide when it will benefit the entire organism. Otherwise, the cooperative falls apart. A condition where there is uncontrolled cell division is called cancer. Let's go over this diagram of typical eukaryotic cell cycle. After a cell has just divided, it is in interphase, or the phase between cell divisions. When it divides, it first separates the copies of its chromosomes so that each of the new cells has a complete duplicate copy. We call this mitosis, and it is part of M phase, or the mitotic phase. Then the cytoplasm of the cell divides. We call this cytokinesis. We can watch M phase and cytokinesis happening under a microscope. We'll go over the stages of M phase or mitosis in following slides. But let's go back to interphase. For a long time, we didn't know what the cell was doing during this time, other than getting bigger. Now, with biochemical technology, we are able to characterize interphase. During G1, or the first gap phase, the cell is growing. It is undergoing cellular respiration to gain ATP to make proteins, lipids, and such. It is responding to its environment by moving through the environment, if it can move, transporting nutrients into the cell, exporting waste out of the cell, and so on. Next is S phase, or synthesis phase. The cell duplicates its DNA to get ready to divide into two new cells. After S phase, or once a complete copy of the genome has been made, the cell makes proteins that it will need to divide. This is called G2, or gap 2 phase. At the end of G2, the cells undergo mitosis and cytokinesis, and new cells result. The cells go into gap 1, and the whole process starts over. Now that we understand interphase a little bit better, let's go over the end of G2 and mitosis in more detail. During interphase, the duplicated chromosomes are unwound, and the genes are being transcribed and translated to make proteins. At this point, the chromosomes are called chromatin. A nucleus is visible, and there are one or two darker areas, called the nucleoli or nucleolus, where parts of ribosome are being made. Two centrosomes have formed by replication of a single centrosome. In animal cells, each centrosome has a centriole pair. Plants have centrosomes without centrioles. As the cell enters M phase or prophase, the chromatin fibers condense into chromosomes, like you see in this picture here. The duplicated chromosomes are called sister chromatids, and they are joined together at several spots, but the only one that we're interested in for this recorded lecture is called the kinetochore. As the chromatin condenses, the nucleolus disappears. This is because the cell stops all production. Think of it this way. When you move, you usually pack everything up. You move boxes to the new place. Until you unpack, you can't find anything, and usually eat out until you can find the pots, pans, and dishes. Well, when a cell divides, it packs up the DNA so it doesn't get broken or otherwise damaged during the move. While it's packed up, the cell can't use any of it until it finishes moving and unpacking. While all this DNA packing is going on, the mitotic spindle forms. It is composed of microtubules which form from the centromere. As the spindle forms, the centrosomes move apart. Eventually they will end up on opposite sides of the cell, also called the poles of the cell. The next phase of mitosis is prometaphase. The nuclear envelope breaks up. The chromosomes are even more condensed, as you can see in this diagram. Microtubules from the mitotic spindle attach to the kinetochores of the sister chromatids. becoming kinetochore microtubules. Microtubules that don't attach to a kinetochore, called non-kinetochore microtubules, cross in the middle. The next phase of mitosis is metaphase. The kinetochore microtubules line up the sister chromatids in the center of the cell. Meta stands for middle, and the chromosomes are in the middle of the cell. There is a kinetochore microtubule on each side of the kinetochore. Anaphase happens next, as the kinetochore microtubules shorten and pull the sister chromatids apart. A copy of each chromosome is drawn to opposite sides of the cell. Meanwhile, the non-kinetochore microtubules kind of push each other as they lengthen, causing the cell to elongate. In telophase, new nuclei form around each set of chromosomes. The chromosomes decondense as the DNA is unpacked. As this is happening, the cytoplasm divides through cytokinesis. When the two cytoplasms separate, two new cells are formed. Let's take a closer look at cytokinesis. In animal cells, a ring of microfilaments or actin filaments forms under the cytoplasm at the equator, where the chromosomes had lined up during metaphase. They tighten and tighten, pulling the cell membrane until it splits the cells. It's kind of like cutting cheese with a wire, but the cell membrane isn't as stiff as a block of cheese. Plants and other organisms with the cell wall can't do this. Plant cells form a cell plate at the equator. Basically, vesicles full of enzymes and supplies for making a new cell wall gather at the equator and fuse. They then get to work making a new cell wall between the two cells. This is more like building a wall in the middle of a big room to make two smaller rooms. Bacteria don't undergo mitosis because they don't have a nucleus that needs to break down and be reformed. Instead, they undergo binary fission. They duplicate their chromosome. Remember, they usually only have one, starting at what's called the origin of replication. As the chromosome is duplicated, the origins move to opposite sides of the cell. This pulls the duplicated chromosomes apart. Then the cell forms a cell membrane and cell wall between the two new cells. The textbook goes into the evolution of mitosis at this point. You can read it. It is quite interesting, but don't worry about studying that for the exam.